All right, so begins our study of genetics. And you'll notice that throughout the notes here, I'm using the Cartoon Guide to Genetics, figures from the Cartoon Guide to Genetics, because it's a great little book and it does a real good job of getting across the main points. And the first point here is that people were trying to figure out how heredity works a long time before Gregor Mendel came along, who is going to be the focus of our study of genetics here at the beginning anyway, because he discovered the fundamental laws of how heredity works. And he's considered the father of genetics. But he was a monk. So monk finds gene, world yawns, and we'll come back to that world yawns in a minute here. But he was the man. He's the father of genetics. And he was a monk in Brunn, Austria. And in his spare time, he bred pea plants in the monastery garden. He was an amateur, gardener for sure, but an amateur scientist also, and a very good scientist. So you'll notice here that the job of discovering how heredity works required superhuman patience, unlimited time, and a miracle of luck. That's what Mendel brought to the study of genetics. Or what I should say is the study of heredity, because at that time they had no idea what genes were, or anything about DNA for that matter. So, of course, because this is the study of genetics, we should start off by defining what genetics is. Genetics is the study of heredity, and we now know that it involves genes, which are sequences of DNA, nucleotide sequences in DNA that code for proteins, which is the whole idea of the central dogma of biology. It's the basis for life on Earth. And again, Mendel didn't know anything about genes. He didn't know anything about what we now consider the central dogma of biology. He was just trying to figure out how heredity worked, how characteristics or traits were passed from parents to offspring. So let's define heredity too. Heredity is the genetic passing of physical or behavioral characteristics from one generation to the next. And again, Mendel didn't know about genetics, but notice the relationship between genetics and heredity. Genetics is the study of heredity, and heredity is the genetic passing of traits from parents to offspring. So they are intertwined. So at the time, in the early 1800s, there were two hypotheses about how heredity worked. The first was known as the blending hypothesis, which suggested that physical traits, what we now refer to as phenotypes, of offspring are an intermediate of the parents, like blending paint. So, for example, if you have a red flower and a white flower, and a red flower plant and a white flower plant, when you cross those plants, when you breed them together, the offspring are going to be pink, just like blending red and white paint. The other hypothesis is known as the particulate hypothesis, which suggested that phenotypic traits can be passed from generation to generation through discrete particles, which keep their ability to be expressed while not always appearing in the descending generation. In other words, these are particles that are passed from parents to offspring, and offspring may receive a particle that doesn't even show up in their phenotype, that doesn't even show up in the way that they look. So even though they have the particle for that trait, they don't show that trait. And of course, I'm using the term particle here, but hopefully you get the connotation that these particles are referring to genes and alleles, if you have any background in genetics at all. And as you can imagine, at the time, the blending hypothesis had a lot of popularity. I mean, it just makes sense that, children seem to be kind of intermediate between their parents. But that's what Mendel was trying to figure out. Is it blending or is it particulate? Or is it some combination of the two? So he grew over 10,000 pea plants in his investigations, and he discovered the fundamental laws of heredity, and they're laws that hold up today. And through his experiments, they were very carefully controlled breeding experiments. He collected meticulous data and applied statistical analysis to that data, which is something that nobody had done before him. And that's really what allowed him to discover the fundamental laws of heredity. And he published his work in 1865. However, as so often happens in science, his discovery, the world didn't really realize what he had discovered. Um, Nobody understood it, probably. And that's where the world yawns comes in here in the cartoon guide. Monk finds gene, world yawns, because nobody realized what he had discovered. And it wasn't until 1900, long after his death, that, well, actually not too long after his death, but after he was dead, that's when some scientists dug up his paper and read it and realized what he had discovered. They understood it and realized what he had discovered. So this caption here ends up with a miracle of luck. And Mendel was extremely lucky in a lot of different ways. And we're going to be looking at each one of those ways as we proceed on here. The first way that Mendel got lucky was in his choice of experimental subject. Turns out peas are perfect for genetic research, as far as plant genetic research anyway, because they have a number of stable varieties that form hybrids, and they're easy to raise and control their breeding, and they produce many offspring. And there are a number of organisms that fit that description, but not all plants have heredity that's as simple as what Mendel observed in pea plants. And today, fruit flies are used pretty heavily for the same reasons. There are a number of different varieties of fruit flies and characteristics in fruit flies or traits in fruit flies. They're easy to raise and control their breeding, and they produce hundreds of offspring. One female fly can lay like 500 eggs. And that becomes important when you're dealing with statistical analysis. And again, that's something that Mendel applied to his results that nobody else had done before. So as we see here in the cartoon, peas have a number of different characters that show only two contrasting traits. So a character, and this is a vocabulary term, of course, for this unit, is a feature that is the expression of a gene, such as height, or seed shape, or pod shape. So the idea here is that the character is height, and the character is height, and here the character is seed shape, and here the character is pod shape, right? So it's like a general description of the result, the possible results in the offspring, or a possible variation in the offspring. And what is a trait? Well, a trait is one specific alternate form of a character, resulting from expression of an alternate form of a gene, which we call an allele. So in this case, again, the character is height, but there are two different contrasting forms of height in pea plants, and that is either tall or short. When it comes to the character of seed shape, there are two contrasting traits. There's either smooth or wrinkled. And in the character of pod shape, the two contrasting traits are plump and pinched. So it's somewhat difficult to wrap your brain around this concept or the difference between character and trait. But that's what I'm trying to emphasize here for you. And then we need to relate that to the genetics that are going on. And these are only three characters that we see in pea plants representatives, but there's a whole list of them here that you can read for yourself. And we're also going to go on to the next slide and take a look at the 7 traits. It turned out that Mendel chose 7 traits to work with, or I'm sorry, 7 characters to work with. And again, Mendel didn't know about genes, but we do. So let's define a gene. A gene is a unit of heredity that's transferred from a parent to offspring that determines a character. So, for example, the character of height, there's a gene that controls height in pea plants. And there's a gene that controls seed shape in pea plants, and a gene that controls pod shape in pea plants. Well, then what's an allele? An allele is one or of 2 or more alternative forms of a gene that determines a specific trait. So in this example, the gene is for height, but there are 2 forms of the gene, one form for tall and one form for short. And those 2 forms of the gene are alleles for that gene. Same for seed shape. The gene is for seed shape, but there's a smooth allele In other words, a smooth form of the gene, and that's the smooth allele, and there's a wrinkled form of the gene, that's the wrinkled allele. And then for pod shape, plump, there's an allele for plump, there's an allele for pinched. So alleles are different forms of a gene. And the final point here is that what you want to do in your mind is equate character with gene. Genes code for characters. Alleles code for traits. So link those in your mind. Genes code characters, and alleles are forms of genes that specify the trait for that character. These are very specific concepts, and you got to be careful in how you use them. And it should become easier the more you use them. So this table shows the 7 characters that Mendel worked with: seed shape, seed color, seed coat, color, pod shape, pod color, flower position, plant height, right? Those are all characters, and they're all coded for by genes. There's a gene that controls each one of these characters. So these are not only the genes that Mendel investigated, the characters that Mendel investigated, they're also the genes that Mendel investigated. But again, he didn't know and didn't call them genes. This also shows his experimental design, at least at the beginning of the experiment. So he repeated the same experimental procedure over very many trials, again, like the 10,000 plants. And he always began with a P cross, what he called the P generation or a P cross, which means a parent that is pure for one trait crossed with a parent that is pure for the contrasting trait. So P stands for parental, but you can also remember that it stands for pure, and that will help you remember that the parents were pure, also known as true breeding. So they were pure or true breeding, meaning that they didn't have the other allele. In other words, this for seed shape, this parent that was round didn't have the allele for wrinkled. Likewise, the parent that was wrinkled didn't have the allele for round. So they were pure for their contrasting traits. And the other thing that was lucky about these traits is that they were either round or wrinkled, nothing in between. Either yellow or green, nothing in between. Even tall or short, nothing in between, no medium, which is, kind of strikes us as strange. But that was lucky, very lucky. Because if there were things in between these traits for each of these characters, then it would have confused Mendel, and he may not have discovered what he discovered. So again, either round or wrinkled, either yellow or green. The second row here shows the offspring from these parents, and you'll notice that it's labeled F1. The F in F1 stands for filial. Filial translates to offspring. So, and the 1 designates that these are the first offspring from the parents, right? Or the first offspring in his experimental design, anyway. So these are symbols and labels for each generation that Mendel used, but you need to be familiar with them, and you need to be familiar with Mendel's experimental design. So, of course, as we learned about scientific investigations and experimentation, you need to repeat the experiment. So even though this shows only 2 parents that are being crossed and only 1 offspring being produced, they actually represent hundreds, because again, Mendel worked with thousands of plants. It's sufficient to show only 1 here in the diagram, though, or in the table of each offspring, because the results were the same every time he did it. No matter how many hundreds or thousands of times he carried out this same experiment where he crossed these same pure parents, the offspring always ended up showing only one trait for each character every time, which again was lucky. Lucky that the heredity or the inheritance of each of these characters worked in what we now consider simple Mendelian way. But of course, to Mendel, these results were surprising because the same trait always appeared for each character, never the other, and nothing in between. We now know this is because those traits are dominant, which is a concept that you're probably already familiar with from studying genetics in previous class. So each of these is a dominant trait. Round's dominant over wrinkle, yellow's dominant over green, tall's dominant over short, and so forth. So how did Mendel do this? Well, to understand what Mendel did, his experimental design, you have to understand something about the structure and function of a flower. So we're going to go ahead and label the important structures that you need to know in order to understand what Mendel did. So the stamen is the structure that at the end of it has the anther, and the anther is what produces pollen, and what's contained in pollen is sperm. So the anther, the stamen and the anther are male reproductive organs in the flower. This whole thing here is called a pistil, and it's made up of a sticky stigma, and that's how you can kind of remember what the stigma does. It's sticky, and that's where the pollen lands when a flower gets pollinated. It sticks to the sticky stigma. The style is simply the stalk that supports the stigma, just like the stamen is the stalk that supports the anther. The ovary is this whole base of the pistil, and then within the ovary are these little ovules, and each ovule produces an egg that, when it gets fertilized at the time of pollination, a pollen tube grows down through the style into the ovary and into each ovule and fertilizes each ovule within the ovary. And those ovules then will develop eventually into seeds. Now, in the case of Mendel and his peas, each pea is a seed, or each of these seeds would develop into a pea. So to summarize, anthers produce pollen and they contain sperm, so it's the male reproductive organ. Ovules produce eggs, and when they're fertilized, they become seeds, or peas in this case. Not all plants or flowering plants have both male and female organs in the same flower. It just so happens that peas do. And when there are both male and female organs on the same plant, that's what we call monoecious. So monoecious means it literally translates to one house. Monoecious, one house. Both male and female reproductive structures on the same plant. And peas are monoecious. And more than that, they have both male and female structures in the same flowers. Some monoecious plants have separate male flowers and female flowers. But in the case of peas, the same flower has both male and female reproductive organs. And that's what it says here in the cartoon guide, both male and female organs in the same plant. Fertilization, as you should all already know, is the fusion of sperm and egg, resulting in a one-celled zygote offspring. So whenever a sperm fertilizes an egg, that's fertilization, and it produces a zygote, which is the one cell that results from fertilization, but that zygote then is going to start to divide and develop into an embryo. When you're dealing with flowering plants, as well as other plants, pollination, you talk about pollination. Pollination is what leads to fertilization. So pollination is the transfer of pollen, and remember, the pollen contains sperm. So it's the transfer of pollen to the stigma of the pistil, and that eventually brings the sperm down to the ovary and the ovules. So the eggs are fertilized within the ovules, which then develop into seeds. But so pollination results in fertilization when we're talking about flowering plants like peas. So now here's where Mendel had a problem, because monoecious plants can self-pollinate. They can pollinate themselves because they have both male and female reproductive organs. They can pollinate themselves. So self-pollination is what Mendel wanted to avoid. It's when monoecious plants pollinate themselves. And this results in less genetic variation. In other words, there's no shuffling of genes between two individuals. It's still sexual reproduction because it involves sperm and eggs, but there's not much variation in the offspring because they have only one parent. So the problem for Mendel is that they fertilize themselves. So what Mendel had to do is figure out a way for the plants to ensure that the plants would cross-pollinate. When an individual plant pollinates another individual plant, a separate, different individual plant. that results in greater genetic variation. But in Mendel's case, it was the only way that he was going to be able to make his experimentation work. If he had to control the mating between two different individual plants. So Mendel wanted to perform cross-pollination in his P generation, his parental generation, so that he could cross plants with contrasting traits and produce hybrids. That was his goal in his experimental design. So as we see in the cartoon here, he wants to practice family planning. And notice he's got scissors in his hands. So we're going to see on the next slide how that plays in. So Mendel's methods, what he did was he started with parent plants by choosing parent plants that he knew were pure for contrasting traits. And again, that's the P generation, and you can remember that P stands for pure or true breeding. He had to force one parent to be the mother by snipping off the immature male anthers. So before the anthers could mature and produce pollen, he snipped them off. He removed the male reproductive organs from the flower. So that flower had to be or became the mother in his experimental crosses, his experimental matings. So in the cartoon guide here, he snipped off the anthers, used his scissors to snip off the anthers. He then used a paintbrush to transfer pollen from the anthers of his intended father to the stigma of the mother. So that's what we're seeing here in the cartoon guide. And that represents cross-pollination. He's cross-pollinating two separate, different individuals. And that's what's being shown here too. Picking up the pollen from anthers and delivering it to the pistil of a female flower. Then he had to make sure that the pollen that he transferred to the female reproductive organs was the only pollen that was ever going to arrive at that pistil. So he put a bag over them. He tied a bag around the mother flower to prevent unintended pollination by insects or wind. And finally, he did all of this to control the parentage of each generation. So this is how he ensured that only the plants that he wanted to mate were mating, that he would cross only the plants that he wanted to cross. And then, kind of cute here, I think the monk is playing God. So that's what Mendel did. Now let's take a look at his results and apply some of the terminology that we need to use to what he observed with each of his crosses. And we're going to use the height gene a lot as an example in this unit. So this is the gene for height. Remember that for the gene codes for the character, and the character here is height. But there are two forms of that character that are controlled by two forms of the gene. So there's the tall allele, which is the form of the gene that codes for tall, and there's the short allele, which is the form of the gene that codes for short. And that, of course, is the parental generation. In other words, Mendel made sure that that tall plant was pure tall and the short plant was pure short, or true breeding tall versus true breeding short. So it would be, reasonable to expect in this cross that between the tall and short plants, it would yield medium height plants, medium height offspring. But that's not what Mendel observed. That would be in support of the blending hypothesis. So if that was what he observed, then again, he might not have discovered what he discovered about the laws of heredity. But that's not what he observed. So showing only two or even one offspring is sufficient here because they were all the same, tall. So, in reality, he did this with hundreds of plants, hundreds of parents, and he got hundreds of offspring. But in this representation here, they're only showing two plants representing the offspring, but it really doesn't matter because they're all tall. They were all hybrids. They were all tall. No matter how many times he did this experiment, it always turned out the same way. So this represents the F1 generation, and those are hybrids of these pure parents. So hybrid is the opposite of pure. Hybrids, but they're tall, so the shortness disappears. and is, it's not evident in the offspring in the F1 generation, all hybrids and all tall. So Mendel saw this and he was surprised because he expected maybe something between tall and short, like medium height plants. But they're all tall. So he's scratching his head trying to figure out what the heck was going on. And so you can imagine trying to figure this out, trying to figure out what was going on without knowing about alleles, without knowing about genes, so he was working from scratch basically. We now know that there is what we refer to as genotype. In other words, the genetic constitution of an individual organism. And you can describe the genotype using either words or symbols. So, for example, homozygous means having two identical alleles of a particular gene. So, for example, the pure parents are homozygous. The pure tall plant is homozygous tall, and the pure short plant is homozygous short. So, pure homozygous dominant, pure homozygous recessive. That's using words to describe the genotype. So, those are the genotype of the parent plants. But we can also use symbols to represent the genotype. So the pure homozygous dominant would be capital T, capital T being for the dominant allele for tall. The short then would be lowercase t, lowercase t. So homozygous dominant, homozygous recessive. Those were the parents in every one of his crosses. One parent was homozygous dominant, the other parent was homozygous recessive, and that gave rise to all offspring that were heterozygous hybrids. And that's the other term, heterozygous, having two different alleles of a particular gene. So, for example, big T, little t. So all the offspring were heterozygous every time he did this experiment with with all 7 of those characters that he studied. All the offspring ended up being heterozygous. So heterozygous describes the genotype in words. Big T, little t describes the heterozygous condition in symbols. One final term then is phenotype. And the phenotype describes how the individuals actually look. In other words, the phenotype of the homozygous dominant parent is tall, and the phenotype of the homozygous recessive parent is short, and the phenotype of all the F1 offspring is tall. They all ended up being tall. That's their phenotype, how they actually look. So we use everyday language to describe that. In this case, either tall or short. In other cases, it would be either round or wrinkled, and so forth.